looking at the idea of the root system as where we zoom into the tip of the root, the transition zone, where some scientists are exploring that idea as like the root brain. And when you think about how extensive root systems are, that each of the tips of each, that whole root system could have this ability to be the root brain idea with signaling. And so this is something that's still really young in thought and experiment. There's a lot of pushback about it. Um, doesn't mean it's wrong. It's just that there's a lot of debate on what this could mean. Is the evidence really supporting any of these ideas or not? And one other, let's see. So last week we were looking at the idea of the action potentials and thinking about movement of neurotransmitter like molecules in the plant and synapses in the root area. So thinking about all of this, it's other neurotransmitter molecules that we find in animals. They're showing up in plants, glutamate, GABA. And then I was trying to show you these images here, herbivory. So the calcium, long distance calcium signals through glutamate-like receptors. And so I didn't realize that I had already made these videos active right in the PowerPoint. I thought and it wasn't showing up on my PowerPoint here, but I wanted to show there's the caterpillar down low, and you can see they labeled the calcium, so it's easy to watch it move through the plant. So right where it's chewing, you can see it really light up, and then it travels through the mustard plant. I believe this is Arabidopsis. And so there's experimental evidence showing these action potentials and neurotransmitters. And then this one, this is where they put glutamate. It's kind of weird. They put glutamate on one part of the plant and then, whoops, come on. Oh, that one's not active. Seriously? Oh, there. And there you can see the whole plant just lights up. So these are ion channels that are opening, binding to glutamate, and then allowing the calcium flow. So it's data like this that they're using to support the idea of some kind of action potential, which is very similar to our brains and how our brains work. And so the debate goes on. There's more research certainly going on, and we'll kind of come back to some of these ideas as we progress through. I think the last bit that we were covering last week was looking at memory and how, you think about how our brains are responsible for our memory. And so, it's kind of a natural progression that they would think about this as well. Whoops. And so we left off around here where they're looking at this in plants and could they have a memory? And so these are the terms that have been used for decades in the plant physiology realm. And really, if you think about it, it's actually kind of describing specific cases of memory. They just loathe to use the word memory for a non-animal organism. So they phrase it in other terms like acclimation or priming. And so if you think about it, though, in different parts of, oh, what's going on there? Has it been doing that before? Is it just, What areas of plant biology have been investigated with regard to memory? Some examples. So the idea of plant herbivore or pathogen interactions. I think I did mention last week that plants can remember what was attacking them previously and use that information to better defend themselves when they're attacked by the same pathogen. That has been shown with fungal pathogenic fungi that attack plants. And with herbivores, the same thing is showing up. So wound responses, they've done some nice studies where they wound the plant and it kicks in the defense responses. And if you wound again, it's a quicker response, which is getting into it probably has a memory it's calling upon if it can respond that much quicker the second time around. They've looked at hormonal um, synthesis by the plant. And so they often see jasmonic acid or jasmonate, that's a plant hormone, and they refer to it as bursts because it's this big upswing in the amount, in the concentration of jasmonic acid. And they see that in particular when herbivores are attacking the plant. And so all of these are getting at the idea that there is a memory response if they are being attacked by the same thing, and you can get all the way down to the physiology of the plant to show, to have data to show and support this memory idea. Epigenetics, we'll get into that a little bit more, but this is the idea that the actual bases of the DNA molecule are not changing. You have something else that is affecting and altering the gene expression besides a mutation event. And so there's certain things like methylating stretches of DNA. We put a methyl group on it. They're looking at small RNA molecules and how those interact with DNA. And we're going to be looking in a short time at this last idea of how epigenetics is allowing for memory into the next generation, where you can have the plant's offspring impacted in such a way that when they germinate and grow, you can tell that they have a epigenetic memory of what their parent experienced. And so we'll look at that a little bit more. It's a pretty in my mind, a pretty wild concept, but there's data that is supporting this idea. With some of the really early work where they were looking at this, the response to light was pretty simple experiment to do back then with Charles Darwin and his son, because of course they didn't have the technology we have currently. So looking at how does a plant respond to light, and they could show that if you give the plant a previous light exposure, then it would kind of prime their cotyledons and it would cause them to direct how they bent toward the light based on the light that they had previously received. And so it's these kind of the acclimation or the priming response going on with response to light. And so plants previously exposed to light were less responsive, but plants that were kept in the dark before being moved towards the light, they stayed and remained in that position for a longer time. And so you have this idea of this conditioned response, which again, they talk about conditioning a lot. When you look at the plant phys literature, they talk about how the plant was conditioned, and yet really that's a memory response is what the more current plant biologists are talking about. And so this, another piece of evidence, they were looking at drought impacts and salty soil, saline stress. And if you give those Arabidopsis, those are those mustard plants, the Arabidopsis plants, if you expose them to drought, and then expose them to drought again, they would more strongly react to that and adapt to that compared to the plants that just didn't get in that. So again, it's kind of like this priming or acclimating the plants to the drought. With saline stress, what they chose to do for that is take seeds and put them in a salty solution. And if you did that before you have them being planted out into the soil, then they would increase their development. It would speed up and they could handle the saline soils. And so that previous exposure, even though it was just the seed, that previous exposure of the seed allowed the plant to germinate and grow that much better when they were put into the salty soil. And so again, if you look at some of this literature, the scientists would say, oh, it's priming, it's acclimation, it's conditioning, but really it is a form of memory if you look at the more broad definition of what memory could be for the plant. Here's a picture from one of the plant phys textbooks, a classic idea here, where if you expose a plant, here they're putting either, it says inducer, whoops, come on, right here. It says inducer, pathogen, or chemical inducer. So you can put like a fungal pathogen on the leaf, and when the fungi attacks, it starts to cause plant death. Well, that's necrosis. And so that action of the fungal pathogen then sends a signal through the plant body to all the other leaves, telling the leaves, you better gear up because I'm getting attacked. You are likely going to be next. So we're not going to get too much into the physiology of it. But basically this SAR, that SAR, that's systemic acquired resistance, which I have written down here. And this is, so systemic means full body. So the whole plant body is gearing up to defend against that particular fungal pathogen. And if you prime the plant beforehand, like you expose them either to the chemical inducer that isolated from the fungal pathogen or the pathogen itself, and then if they get exposed to that a second time, the exact same fungal pathogen, they're going to react that much more strongly. And so they're actually looking at using these concepts when they're planting agricultural crops. Like, could that be a strategy to get the plants in the field? So the idea here is, and they're getting into some pretty fun technology, but basically, if they can tell that the plants are going to be getting attacked, which they're using with highly sensitive sensors, then the growers can release a certain chemical out into the field that tells the plants, hey, gear up. And so you can get your plants doing the systemic acquired resistance even before the fungal pathogen has taken hold in that field. And so they're really taking this knowledge and trying to think about how could we grow healthier plants that are going to make it to harvest and not get succumbed to this fungal pathogen. And so if you are a grower and want to harvest your plants, you want that whole plant to be immune to that fungal pathogen in this case. And so this knowledge is really important applications too. There's other hormones involved. It's not just a set one or two. They are also exploring salicylic acid, which is related to the chemical acetylsalicylic acid, which is fun to say. So do you remember or do you know what acetylsalicylic acid might be? Something that we can take. Yeah, it's aspirin. So they discovered salicylic acid is a plant hormone. And so we'll come back to plant hormones as we think more about their behaviors. But so we have salicylic acid involved in this. We have the whole body response. You have to signal. So that signal transduction, this is the idea of there's chemicals, could be action potentials flowing through the plant to have the plant gear up. Jasmonic acid and ethylene are also involved. So again, it's just not one hormone. There's usually a suite of hormones that will be involved in this response. And then even with the herbivore itself, the plant is getting clues from that. Um, so when certain herbivores chew on certain parts of the plant, it will give off certain plant volatiles, depending on what part of the plant body is getting chewed on. And that can then trigger the plant to do those jasmonic acid bursts. GJA is jasmonic acid bursts. And that especially is happening if it's the same herbivore coming back, after a time period. And so if it's continually getting attacked by the same herbivore, you tend to see these bursts of jasmonic acid, which help the plant gear up its defense genes. It turns on the transcription of those genes to help the plant defend itself. And again, it's a form of memory. Once the herbivore comes back and starts chewing again, the plant is geared up and ready to go that much quicker. And so they call that a type of memory. Even when herbivores might be laying their eggs on plants, that's a very common strategy. You get all kinds of creepy crawlies that will do that. And they took some tomato plants and ran it through with eggs being laid on the tomato plants and on the leaves. And they could tell just that was enough to kick in their plant defenses. And whoops, they responded more effectively against the grown-up, once the egg hatched. What do you call it with insects? I don't know if they use the word hatch, but once it became the caterpillar, the plant was able to defend itself that much quicker. And so again, this idea of memory. So then, of course, kind of gets you thinking about, well, okay, is this like short-term memory, long-term memory? Can we look at that in any way? And so they've been exploring this idea, and at least so far, the data is showing that when they do the herbivore studies usually they're doing caterpillars for that, you can see the plant being able to remember anywhere from hours to days, depending on the study design and the plant species. There was at least one study where they said it was the life of the plant, but we're talking a shorter growing season type plant. So it was tomato. And so tomatoes, usually grow for several months and that's pretty much it. So they had the seeds primed. They exposed the seeds to a chemical that was going to gear up the tomato against the herbivores and the pathogens that they then exposed the plant to. And it lasted 8 weeks for that particular experiment. On the other hand, impressively, they did a study with birch trees, and this one was a lot longer process. So they did the phase 1 part of the research where they exposed the birch trees, and then 5 years later, that's some patience, they came back and, compared to the controls, the birch trees that were primed 5 years prior, they were able to respond more quickly and more strongly to that particular caterpillar that they had been exposed to 5 years ago. So that study was pretty impressive. They're wondering, especially with the birch study, because it was a 5-year gap and it was still able to respond. like that. They're wondering if there were proteins that were somehow the proteins were made during the first exposure that lingered, that stayed with the plant and was able to quickly like alter gene expression. So like a transcription factors that were able to um, alter very quickly gene expression when they got attacked again. They're still looking at all of this in more detail. Okay, so this idea of memory and neuronal-like activity, synapses, neurotransmitters. Um, where is the... Well, let me go through this one first. So here they're looking at physical contact. So if you are a caterpillar or a moth and you're on a tomato plant and you're walking across it, if you've ever looked carefully at tomato leaves, they have trichomes, those hairs on the surface, and that can the action of the insect walking across and chewing across, it can rupture the trichomes on the tomato. And so, but just the physical contact against the trichomes caused the trichomes to rupture, and then that triggered the defense genes of the plant. And when they were running this study long term, they saw that the plant would actually start to make leaves, the new leaves that it was growing would end up with more trichomes. And so the influence of the insect in, the lifespan of that plant as the plant was continuing to grow was altering how the new leaves that were growing, how they would look and the number of trichomes on them. So that was an additional part of the study that they thought was really interesting, how the plant adapted and responded so quickly like that with the new leaf growth and more numerous trichomes. This one was fun. They took pea tendrils and they figured out pea tendrils need blue quality light in order for them to coil around a support. Plus, they need the physical contact of the support. They need to feel that. So the two things, blue light and mechanical touch. So what they did, they exposed the pea tendrils and gave them either blue light or touch, not both. And then they looked to see if you do like blue light first and then wait a few hours and then do touch or vice versa, how will they behave? And normally when you look at pea tendrils, when they're outgrowing in your garden or whatever, they will grow and start coiling during daylight hours. That's what they're doing. And that's just their normal pattern. But if you stick the pea plants in the dark, they don't coil. So, I mean, it's a daytime thing. And of course, sunlight has blue quality light as part of sunlight. So if you keep them in the dark, they're not going to coil because there's no blue light. And if you don't give them anything to any burst of light or anything, you do a totally dark, like a girl's chamber. And then if you introduce a stake or some kind of suitable support for the pea tendril, they won't coil because they haven't had any of that blue light. And so that was where they were showing, okay, yep, plants definitely need blue light. And then if they took pea plants at night and touched them, and what they did was they touched one side of the pea tendrils. If they touched them, they wouldn't coil at night. But then when you expose them to light after several hours, they could remember that they had been touched. Now when they're exposed to the light, then they start to coil. So again, they're pointing to these experiments that, in this case, the pea tendril was remembering that it was touched during the night. And once it got exposed to blue light, it went ahead and coiled. And so again, showing the memory impact here. So pretty simple experiments. But again, if certain plant physiologists in particular would say, oh, that's just conditioning, or oh, that's priming. And so just be aware of that. You can do this with cold conditions too. You can expose your plants to cold, and then they'll acclimate and get used to that if you do it slowly enough. If you take rye plants, they call them naive if they've not been exposed. And so you take naive rye plants and then you go ahead and expose them to -5, they're going to freeze. But if you took plants that had been previously exposed for short amounts of time, acclimated, then they could go all the way down to -30. And so again, this acclimation idea, but when you think about the broader aspect of memory, how we define memory, it fits that too. And so there's been a lot of data out there on memory, plant memory. It's just been called different things. And a lot of plant physiologists want to keep it that way. They don't want this idea of plant memory. They're not comfortable with it. And so just be aware, there's definitely debate about this idea. Okay, I want to shift to a video before we get into some of the other. Let's see if I can. This is the one. No. All right, let's see if I can get this to work.
Co-founder of Backyard Brains, and our mission is to train the next generation of neuroscientists by taking graduate level neuroscience research equipment and making it available for kids in middle schools and high schools. And so when we go into the classroom, one way to get them thinking about the brain, which is very complex, is to ask them a very simple question about neuroscience, and that is, what has a brain? When we ask that, students will instantly tell you that their cat or dog has a brain, and most will say that a mouse or even a small insect has a brain, but almost nobody says that a plant or a tree or a shrub has a brain. And so when you push, and because this could actually help describe a little bit how the brain actually functions, so you push and say, well, what is it that makes living things have brains versus not? And often they'll come back with the classification that things that move tend to have brains. And that's absolutely correct. And our nervous system evolved because it is electrical, it's fast. So we can quickly respond to stimuli in the world and move if we need to. But you can go back and push back on students. You say, well, you say that plants don't have brains, but plants do move. Anyone who's grown a plant has noticed that the plant will move and face the sun. But they'll say, well, that's a slow movement, that doesn't count. So that could be a chemical process. But what about fast moving plants? Now, in 1760, Arthur Dobbs, the royal governor of North Carolina, made a pretty fascinating discovery. In the swamps behind his house, he found a plant that would spring shut every time a bug would fall in between it. And he called this plant the fly trap. And within a decade, it made its way over to Europe, where eventually the great Charles Darwin got to study this plant, and this plant absolutely blew him away. He called it the most wonderful plant in the world. This is a plant that was an evolutionary wonder. This is a plant that moves quickly, which is rare. It's a plant that's carnivorous, which is also rare. This is the same plant, but I'm here today to tell you that's not even the coolest thing about this plant. The coolest thing is that the plant can count. So in order to show that, we have to get some vocabulary out of the way. So I'm going to do what we do in the classroom with students. We're going to do an experiment on electrophysiology, which is the recording of the body's electrical signals, either coming from neurons or from muscles. And I'm putting some electrodes here on my wrists, and as I hook them up, we're going to be able to see a signal on the screen here. And this signal may be familiar to you. It's called the EKG, or the electrocardiogram. And this is coming from neurons in my heart that are firing what's called action potentials. Potential meaning voltage, and action meaning it moves quickly up and down, which causes my heart to fire, which then causes the signal that you see here. And so I want you to remember the shape of what we'll be looking at right here, because this is going to be important. So this is a way that the brain encodes information in the form of an action potential. So now let's turn to some plants. So I'm going to first introduce you to the mimosa, not the drink, but the Mimosa pudica. And this is a plant that's found in Central America and South America, and it has behaviors. And the first behavior I'm going to show you is if I touch the leaves here, you get to see that the leaves tend to curl up.
The Acrobat Studio is only one PDF spaces with AI do. Drag and research, drop and solve. AI pull insights from your docs. You did your thing. It's time to share and let your team build.
There's no way to bypass this, is there?
Do that with the all-new Acrobat.
All right.
And then the second behavior is if I tap the leaf, the entire branch seems to fall down. So why does it do that? It's not really known to science. One of the reasons why could be that it scares away insects or it looks less appealing to herbivores. But how does it do that? Now that's interesting. We can do an experiment to find out. So what we're going to do now, just like I recorded the electrical potential from my body, we're going to record the electrical potential from this plant right here, this mimosa. And so what we're going to do is I've got a wire wrapped around the stem, and I've got the ground electrode where? In the ground. That's electrical engineering joke. All right. So all right. So I'm going to go ahead and tap the leaf here, and I want you to look at the electrical recording that we're going to see inside the plant. Whoa, it is so big. I got to scale it down. All right. So what is that? That is an action potential that's happening inside the plant. Why was it happening? Because it wanted to move, right? And so when I hit the touch receptors, it sent a voltage all the way down to the end of the stem, which caused it to move. And now in our arms, we would move our muscles, but in the plant doesn't have muscles. What it has are water inside the cells. And when the voltage hits it, it opens up, releases the water, changes the shape of the cells, and the leaf falls. Okay, so here we see an action potential encoding information to move, all right? But can it do more? So let's go to find out. We're going to go to our good friend, the Venus flytrap here, and we're going to take a look at what happens inside the leaf when a fly lands on here. So I'm going to pretend to be a fly right now. And here's my Venus flytrap. And inside the leaf, you're going to notice that there are 3 little hairs here, and those are trigger hairs. And so when a fly lands, I'm going to touch one of the hairs right now. Ready? 1, 2, 3. What do we get? We get a beautiful action potential. However, the fly trap doesn't close. And to understand why that is, we need to know a little bit more about the behavior of the fly trap. Number one is that it takes a long time to open the traps back up, about 24 to 48 hours if there's no fly inside of it. And so it takes a lot of energy. And number two, it doesn't need to eat that many flies throughout the year. It only needs to eat a handful. It gets most of its energy from the sun. It's just trying to replace some nutrients in the ground with flies. And the third thing is it only closes the traps a handful of times until that trap dies. So therefore, it wants to make really darn sure that there's a meal inside of it before the fly trap snaps shut. So how does it do that? It counts the number of seconds between successive touching of those hairs. And so the idea is that there's a high probability if there's a fly inside there, they're going to be put together. And so when it gets the first action potential, it starts counting 1, 2, and if it gets to 20 and it doesn't fire again, then it's not going to close. But if it does it within there, then the fly trap will close. So we're gonna go back now. I'm gonna touch the Venus flytrap again. I've been talking for more than 20 seconds. So we can see what happens when I touch the hair a second time. So what do we get? We get a second action potential, but again, the leaf doesn't close. So now if I go back in there, and then if I'm a fly moving around, I'm gonna be touching the leaf a few times. So I'm gonna go and brush it a few times. And immediately the fly trap closes. So here we're seeing the fly trap actually doing a computation. It's determining if there's a fly inside the trap, and then it closes. So let's go back to our original question. Do plants have brains? Well, the answer is no. There's no brains in here. There's not a, no axons, no neurons. It doesn't get depressed. It doesn't want to know what the Tiger score is. It doesn't have self-actualization problems. But what it does have is something that's very similar to us, which is the ability to communicate using electricity. It just uses slightly different ions than we do, but it's actually doing the same thing. So just to show you the ubiquitous nature of these action potentials, we saw it in the Venus flytrap. We've seen an action potential in the mimosa. We've even seen an action potential in a human. Now, this is the euro of the brain. It's the way that all information is passed. And so what we can do is we can use those action potentials to pass information between species of plants. And so this is our interspecies plant-to-plant communicator. And what we've done, we've created a brand new experiment where we're going to record the action potential from a Venus flytrap, and we're going to send it into the sensitive mimosa. So I want you to recall what happens when we touch the leaves of the mimosa. It has touch receptors that are sending that information back down in the form of an action potential. And so what would happen if we took the action potential from the Venus flytrap and sent it into all the stems of the mimosa? We should be able to create the behavior of the mimosas without actually touching it ourselves. And so if you allow me, I'm going to go ahead and trigger this mimosa right now by touching on the hairs of the Venus flytrap. So we're going to send information about touch from one plant to another. So there you see it. So I hope you learned a little bit something about plants today. And not only that, you learned that plants can be used to help teach neuroscience and bring along the neuro revolution. Thank you.
What'd you think? Whoops. What did he call it? Interspecies communication?
PDF spaces with AI.
Ugh. Is it just me, or have they inserted way more ads into YouTube videos? That video that I've showed you didn't used to have any interruptions, and now all the ads. So action potentials, going back to his video. So he's a neuroscientist, and he's using plants to teach about neuroscience. I wonder if they do that here with our neuroscience classes. Anyone taken neuroscience here? I should give them a heads up. They might want to do that. So action potentials in plants, even being able to do that from the Venus flytrap over to the mimosa. Pretty cool. All right. I can post that video in Educat. I don't think I did that one yet. I did the other one, or maybe I did. Ugh. All right. All right. Oh, my computer. Okay. I have no idea what that's about. Okay. All right. So I wanted to show that, well, for several reasons. One is I wanted you to see how the mimosa behaved when it got disturbed. So the action of it closing its leaflets from fully expanded into folding up, and then the whole leaf drops down as well. And somewhere I can't find it lately. I keep looking for it. If anyone finds it, let me know. But I think it was a Sir David Attenborough video clip where he showed like a insect crawling on the mimosa, and you can see right where it's crawling that it closes up its leaflets, and then the beetle manages to stay on, but then when the whole leaf bends down, the beetle like rolls off and onto the ground. And it's put to really cute music. And Sir David Attenborough says something really pithy about, that it's a confused beetle now. It doesn't know what happened. And you see the beetle, wander off and stay off the mimosa from that point forward. So they do think that that action is to prevent herbivory. They do think that that's an effective mechanism. But a group of scientists said that plant would be a great study plant in order to look at memory in plants. And so this is one of the first papers published where they did some really rigorous studies in a current era. So this is 2014. So this is one of the landmark papers on memory in plants. And Monica Gagliano was the first author, but you see Stefano Mancuso is the last author. All of these scientists are in the plant neurobiology realm, which has been renamed to plant signaling and behavior. But basically, they wanted to see if plants could have a memory. With mimosa, they were looking at a trade-off between predation risk or having to get access to light. So obviously a plant needs light to do photosynthesis and stay alive. On the other hand, it doesn't want to get eaten. And so there's this, you have to think about both if you're a plant. And I love the last phrase there, a context that is ecologically relevant to this plant. And so sometimes you have to really look at the experimental design, because I think some experiments are set up in a way that it's really not challenging the plant in an ecologically relevant context. So you really need to think about that. So I think they did an excellent job with how they did this experiment, and really thought through it carefully when they did their experimental design. So this is their definition of what memory is and learning. Learning is a process of acquiring memories by which adaptive changes in an organism's behavior arise as a result of experience. Okay, so they quoted that right in the paper. All right. Another definition is what is habituation. It's an extremely important or adaptive aspect of life. Thanks to it, organisms learn to pay attention to stimuli that are truly meaningful in their environment while ignoring those that have proven irrelevant or innocuous. Okay, so truly meaningful in their environment. So again, you got to look from the plant's perspective. What could that be? So this is from their paper, how they're showing their experimental design. So they had this support where they connected a pot onto it, and then they dropped the plant 15 centimeters into a foam base so that it wouldn't bounce when it hit. So it would drop and just stop. Okay, drop and stop. And they would take data before they drop. They'd measure the diameter or width of the leaf, and then they'd measure again after the drop. So that was the chunk of data that they were using. And then they would drop the same mimosa plant over and over and over. And they say in here, well, they don't really say in there. The setup ensured that a standard level of disturbance was administered to all plants, and it was sufficient to force the closure of all leaves. All right. So they would make sure, they had to first make sure that this would make them close their leaves. All right. And then the actual experiment was, okay, now we're going to teach the plants that they don't have to close their leaves, essentially, that this is not relevant to them. And so they would then start to drop the plants during their experiment a set amount of times over and over and over and over. And then they had something that they called dishabituation as part of their experimental design. So they were thinking this through and they're like, well, if it takes energy for the mimosa to be doing that. And if they just get fatigued, maybe that's why they're going to just close their leaves and keep them closed. And it's really due to fatigue and they're not really learning anything. And so they said, let's do a dishabituation part to this experiment where they did another disturbance to mimosa that was different than drop. So what they did was they shook the plant to prove, to support the idea that they're not fatigued. They're still going to respond to that. It's not due to fatigue that they're behaving in a certain way. So this is essentially what they did. Oh, let's see if I can get it there. So they did it, well, that's at 225 RPM. They did theirs at 250. Okay, so it's even faster than that. So that's a shaking table where you shake at a certain revolution per minute. And so they did that to the mimosa as the dishabituation to prove that they can respond to this. They're not too fatigued. They can do this. And so then they ran the experiment and um, in a single training day, so in one day, each plant received the first train of 60 drops at time 0, and then the second train at 10 minutes later, another 60 drops. That's a lot of dropping. They really went for it. Um, a sufficient intertrain interval, the 10 minutes, to allow all plants to recover and fully reopen their leaves. Subsequent training sessions were then delivered 1, 2, 4, and 6 hours later. And then they did the dishabituation to show that they're not too fatigued. That's not why they're behaving that way. And then they were also looking at low light versus high light. So if you are a mimosa plant and you're being exposed to low light and you're pretty much a high light plant, you like lots of light. So if you're a mimosa and you're put into low light, you're probably going to be worried, so to speak. You're probably like, oh boy, I really need to keep my leaves open and flat and just sucking up that sunlight as much as possible. All right. And so they did a low light LL and a highlight where the other group of plants were in their typical highlight that they prefer. And you can see that I mean, it's amazing at just even you look from 0 minutes to 10 minutes after the 60 drops, they reopen their leaves and you can see there's hardly any change in their leaf diameter essentially with subsequent training sessions. But they did respond during dishabituation. So when you put them on the platform shaker and shook them, they closed up on that because that was a novel experience to them. And so they definitely closed up, and that was trying to show they're not fatigued. They're choosing to keep their leaves open, even though we drop them 60 times. All right. And then the low light you can see are even better at maintaining more openness to their leaves because they're really like, I want that sunlight and I'm going to stay open as far as I can to get as much access to that sunlight. Does that make sense? Okay. So really well designed. And really, they were quite stunned at how quickly the plants were reopening. And so even after the first 4 to 6 drops, they didn't have to wait to 60 drops. After like 5 times, the plants are like, this isn't relevant. They were learning this isn't relevant to me. It's okay to keep my leaves open. And so The leaf folding reflex habituated more rapidly and leaves reopen more fully under low light. The result shows that a greater ability to ignore a recurrent yet harmless stimulus being dropped like that in order to minimize energy waste and optimize opportunities to forage for light. So the maintenance of the habituated response despite dishabituation. So what they mean here is after you dishabituated them and shook them on the rotating platform, they bounce back. So they're saying that's what they're pointing to on this with this statement. Confirms that the response decrease cannot be attributed to fatigue or sensory adaptation. Instead, it denotes the selective nature of habituation, an active learning process whereby plants, like animals, perceive an innocuous stimulus but choose not to respond to it while still remaining responsive to the surrounding environment. What we have shown here leads to one clear, albeit quite different conclusion. The process of remembering may not require the conventional neural networks and pathways of animals. Brains and neurons are just one possible, undeniably sophisticated solution, but they may not be a necessary requirement for learning. So that's a quote taken from their conclusions. So again, really well-designed study coming at it from different angles to really firm up their experimental design and show that at least with the mimosa plant, it learned very quickly to ignore innocuous stimulus. Like, okay, that doesn't really matter that I'm being dropped. It's not ecologically relevant to me. What I want is light and as much of it as I can. So I'm going to keep my leaves open. So this was published back in 2014, and when it came out, it met with criticism. So this is one of the papers that was written in response to Gagliano's group. Insufficient evidence for habituation in mimosa. And so I think I posted this too in the readings if you want to look at it. But basically, Robert Biegler was not convinced of mimosa being able to learn to ignore that innocuous stimulus. And so that was in 2018. So her paper was published in 2014. He wrote a rebuttal 4 years later. So then they wrote a rebuttal to that rebuttal. Plants learn and remember. Let's get used to it. I love it. I just love it. She's got a sense of humor. But anyways, so this was 2017. They must have been in conversation behind the scenes. Anyways, in that paper, they say, clearly, the best challenge for these findings is further empirical work. In addition, while we feel that Biegler offers some good suggestions on how to further improve investigations of mimosa's habituation, it is a little disappointing to see no experimental data accompanying his arguments, especially given that Gagliano's study was published some time ago. So in his rebuttal, he really didn't come up with a better experiment and say, no, this is what's really happening. He didn't do that. And so they're criticizing that. Finally, after some initial defensive reactions to the perceived danger of considering plants much more capable of performing cognitive feats, learning, memory, decision-making, problem-solving, and more, then we would like to acknowledge perhaps the time has come to finally get used to it. And so she's not afraid to get to the combat field and push back. So so Gagliano and several scientists that she routinely collaborates with and does research with are working very hard in this realm of plant biology, looking at all, the learning, the memory, the decision making, problem solving. And so we will be going through some of these ideas in this concept of plant behavior in this class. So I would like you to be comfortable as you're studying with thinking about that experiment. You don't have to get too far into the rebuttal. You don't have to read the rebuttal paper or her response to that, but the actual paper, what's going on in there. So, I mean, I covered the highlights with you here. That's what I'm expecting you to know, but use the paper as the reading to come up with your thoughts on what's going on with that. Any questions on that experiment or that concept? Did it convince you? Plants can learn? So that TED Talk we just watched, he did kind of touch on this, how do plants move without muscles? And I think that's important to think about because some of the behavior we're talking about is a movement of some sort that we can see with the naked eye. And so basically, when you think about a plant that doesn't have muscles, no skeletal system, how is it moving? There's two main ways. It's water pressure. So turgor movements means it's in response to water pressure changes. And then growth movements. The actual growth of the plant is a movement in and of itself. So what he was talking about in that TED Talk with mimosa, he was saying that it's hydrostatic pressure shifts going on. And so if you look at the design of their leaflets and their leaf, I think I mentioned this briefly last week, it's the pulvinous region. So that's just a tissue region of the plant. You can see it's happening. You can find it at the base of each leaflet. There's pulvini. And then at the base of the leaf itself, there's a pulvinus. And what they're trying to show on this left diagram is that in the pulvinous region, there's this shift in turgor pressure, is I guess how I like to think of it. And so you see that this region of the pulvinus down here is losing turgor pressure. So water is rushing out of that, whereas water is rushing into this region of the pulvinus and causing turgor pressure. So turgor just means you've got water pressure pressing against the cell membrane and the cell walls in the pulvinous region in this particular case. This diagram is also showing a little bit of the pulvinous tissue. They call it the motor organ here, but that's the pulvinus. And here's a blow up where they're showing this side of the pulvinus is losing water, so it's becoming more flaccid. It's losing pressure. And then this side is gaining pressure. And so that shifting in where you see less pressure and more pressure causes that movement of the leaflets and the leaf. And it can be quite rapid. The plants can shift their water in the pulvinous region quite rapidly. And so that's why you can see it so readily when he was touching the leaves, they immediately reacted to that. So the mimosa it's really a nice model organism to look at that idea. Also, though, all the plants that you can think of pretty much have stomata, and that's what you're seeing in the lower images. So on the leaf, you have stomata that are composed of these guard cells that surround a pore. All right. So you have guard cells and a pore. And depending on what's going on in the plant, they can have their stomata wide open, like you see on the left, or this one is more closed down. And so plants are able to regulate the flow of water in and out of their guard cells. All right. So the guard cells can get shifting water in and out, and it's actually the case when water rushes into the guard cells, it makes the pore more open in the middle. When water rushes out of the guard cells, it makes the guard cells go flaccid and it covers over the pore in the middle. And so you can see that happening on a minute by minute basis sometimes when plants are out in their native habitats and they're dealing with their environment. You have this trade-off where when a plant opens up their stomata, immediately water vapor starts coming out. And so they're losing water as soon as they open their stomata. And sometimes that's okay, and sometimes that's not so okay. If they're going through a drought, they don't want to dehydrate, and so they have to regulate the size of their pore quite closely. When they open their pore, of course, now they can get access to CO2. CO2 is coming in that way, and so the regulation of their stomata, their guard cells and the stomata are really, really important, and it can be very rapid. So turgor movements again are helping get the guard cells right where they need to be, whether it's 100% open for the pore or 10%. And so, whoops, it is, some very special cases of turgor movement that you can see. The Venus flytrap also has some turgor movements going on too at the base of the leaf. So those are pretty dramatic. You can see that in real time. Growth movements sometimes are take a while to show up, but if you think about the plants, how they grow, it's at their apex, the stem tips, the root tips that you see new cells dividing and growing and giving length to the roots, length to the stems. And so this process of cell division, creating new cells that adds length, and then also don't forget that cells or plants cells can grow by just getting bigger. And there's a finite size, but you can see that cells do elongate in the plant body. And so this is often you can see this growth response. It can be different depending on what part of the body you're looking at. And so, if roots are trying to grow toward gravity, we call that positive gravitropism. If the shoot and leaves are trying to grow away from gravity, then we call it, anti-gravitropism. And so you have these ideas that you'll get different growth rates, especially when you look at the typical plant body where you have the shoots and leaves above the soil, the roots below. Those are two very, very different environments that have different requirements. And so you'll often see different growth rates, especially when you compare the above versus the below ground. So moving with growth movements a little bit slower unless you use special videography where you can speed it up. We talked a little bit, I think, about phototropism, or it's the idea that Darwin and his son were looking at how a plant grows toward unidirectional light. And so phototropism is a growth response. That's actually a growth response. That's not turgor pressure shifting going on here. And so those coleoptils, those little baby plants that are being exposed to unidirectional light, they are starting to bend toward that. And they did figure out later, not Charles and his son, but later with further research, they found it's the blue quality of light that's causing the blue quality coming from sun is actually causing the bending pattern toward that unidirectional light. So in this case, if you're above ground and the coleoptils, by the way, are photosynthetic, they're green and photosynthetic. And so think of it as a baby seedling. And so they're trying to orient their photosynthetic tissues to be pretty much like 90 degrees to the incoming sunlight to maximize photosynthesis. The typical plant tries to get their leaves 90 degrees to wherever the sun is in the sky, and that gives them maximum photosynthetic rates. On the other hand, you can think about gravitropism, and when you think about roots, they want to grow toward gravity. And so again, this is a growth response. They're going to grow. So they're going to have cell division and growth toward gravity. And they actually do that. Oh, I forgot. Let me go back. with phototropism, what's happening here is that that blue quality light is striking the above ground portion of the plants. And to get that bending growth response, what actually happens is the hormone auxin is shifting. It's produced in the tip and it starts to shift down the unlit side or the dark side of the baby plant and stimulates that side of the plant to grow. So you have asymmetrical growth. In this case, the left side is getting stimulated, grow, grow, grow. The side that's getting lit by light is being told, don't grow so much. And that's what's causing this bending pattern toward the light. Okay. So with phototropism, auxin is actually stimulating the plant to grow toward the light. With gravitropism, you have a weird situation where auxin, IAA, that's auxin, it's actually weird because down in the roots, what's happening is if you take a root and it's growing straight down toward gravity, you can see auxin comes down the root. And what they're trying to show is that auxin flows down to the root tip in A and then kind of flows back up. They call it the auxin fountain or something like that. And so it comes down the root and then equally on both sides flows up. All right. If you take a root and put it horizontal to the ground, it's perceiving gravity is, down. And so what actually happens is auxin comes down to the root tip and then you can see this uneven distribution of auxin. The different size arrows means different amount of auxin. You can see more auxin is on the lower side of the root half, less auxin on the top. Okay, this is weird because I just told you with phototropism, auxin speeds up cell division and gets the plant to bend. In the roots, it does just the opposite. If you have a lot of auxin in the roots, instead of speeding up the root, it does the opposite. It's like slow down, slow down, slow down, slow down your cell division and growth. Less auxin actually speeds it up. So auxin gives two very different signals, depending on if it's happening in the above ground or the below ground in this case. So just be aware that sometimes you can't just say this hormone does this for the plant. And you often have to think, well, does it do it for the whole plant? Or is it doing something different for the above versus below ground? All right, so that shift in the auxin causes the roots to start to grow. So with less auxin, that's actually stimulating the above section of the root to grow, 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 grow. And as it does that, it's going to start bending down to that asymmetrical growth. They've been wondering how the plant is sensing gravity, by the way. It's the amyloplasts. So amyloplasts, if you plant kingdom, they're just plastids that store starch. And so roots have a lot of amyloplasts. And so they had to give them a fancy name, stadoliths. So in the root tip area, they say it's these particular amyloplasts that they decided to call stadoliths that are actually perceiving the force of gravity. And so here you can see in figure A, all the stadoliths are at the bottom of this particular cell, kind of even. But if you tip roots onto its horizontal side like that, you can see how the stadoliths shift their position. And they think that causes a different pressure against the cell membrane compared to if it's straight down. And that little difference is enough where it can figure out, oh, gravity, and they can grow toward gravity. Okay. Okay, let's take a break. I forgot to give you a break. Let's take a break for a little bit here, so you can get up and walk around and wake up a little bit. And then we'll come back to this. Sorry, forgot to give you. Okay. All right, so movement. You got turgor movements, growth movements. Sometimes it's an asymmetrical growth that on either side of the tissue or above ground versus below ground. One other concept I want you to keep in mind is thigmomorphogenesis, which is a mouthful. Thigmomorphogenesis. Thigmo means touch. Morphogenesis means a change to create change. Morphology is referring to body, genesis to change. All right. So touch that creates a change in the body form of the plant. Change in overall growth pattern due to mechanical disturbance. That's another way that it's often defined. So in this case, the mechanical disturbance was just someone coming along and physically running their hand on over the shoots and leaves of the left plant. The right one was left alone, undisturbed. And I think they did this 2 to 3 times a day. That's all. And so they touched that left one 2 to 3 times a day, and you can see a huge difference in the overall morphology of the plant. Same exact species. It started out at the same height and everything, and you can see a big shift. And so this idea that touch can have a profound impact on the plant development is something that you need to keep in mind. And when they first started looking into this idea of can plants sense touch, the animals, they already had a lot of that figured out. They were surprised to find that when they did the work with plants, they, at least at the cellular level, the physiological response is quite rapid. And you can see all kinds of things happening. But because plants grow on a different time scale than us, we tend to see any obvious differences that happens it takes quite a while for that to happen. And so again, videography would help with that. If you speed it up, video over time and then speed it up, you would be able to see the change in this case. But when they were doing research on this idea, wasn't Janet Bram was actually not doing a touch study. She was actually doing work with hormones. She was looking at gibberellic acid and spraying the plants with a solution of gibberellic acid. And usually when we do this in plant physics experiments, we usually have like dwarf pea plants that can respond to gibberellic acid, but they typically grow really short. But if you spray them with gibberellic acid, they really grow quite rapidly and quite tall. And so she was doing this kind of experiment where she had a spray bottle with gibberellic acid mixed in with water, and she was spraying. But I can't remember if she worked with pea plants or a different species, but when she had her control had water, she had a spray bottle, a control spray bottle that just had water, and then the one that had gibberellic acid and water. And when she was spraying the control onto the plants, it was impacting them the same at the genetic level. The same genes got turned on. And so she's like, well, wait a minute, gibberellic acid, the presence of gibberellic acid should be doing something. How come they're both responding the same way? And her conclusion was that, well, they're both getting touched by what's in the spray bottle. When she sprays those onto the plants, the droplets are from both conditions, the control and the spray bottle with gibberellic acid are, it's the same idea. And so the eureka moment when she realized the common factor in the treatment was the physical sensation of being sprayed with the solutions. And so that was kind of they point to that as serendipitous because it did really start a lot of scientists going, oh, okay, so maybe plants are more responsive to touch than we thought. And so they started looking into that more deeply, and they found what they call touch genes, TCH genes, which they generally just say touch genes. And they saw that these genes, when they get turned on, they code for proteins that are involved in calcium signaling, which we've talked about a little bit. We know that plants can ramp up their calcium and use it as signaling during action potentials. And so when they started looking more carefully at calcium, the plant physiologist like, okay, what is calcium doing for the plant? We know plants need a lot of calcium. If you deny plants calcium, boy, they don't do well. And they often, I've done those experiments, and the plants get they hardly grow at all. Their leaves are very crumpled and distorted. They just look like a hot mess. And so they've been studying what calcium is all doing for the plant. It's involved in turgor pressure. It's important for that. Electrical signaling, which we've covered. The cell-to-cell communication, which we've thought about. They call it a second messenger because it's involved in signal transduction. When a plant is assessing its environment and then interpreting that and then telling the cells what to do at the genetic level, that's the idea of signal transduction, and calcium's heavily involved in that in order to get that signal brought all the way down into the nucleus and to the level of the DNA. It's even important as a structural component. So it's not always just a chemical used for signaling, it's actually part of the cell wall. And if you deny that, the cell wall does not form correctly. So these touch genes, the calcium signaling, what they found is that the calcium needs to interact with these binding proteins that, so it's like this complex. The calcium binds to the protein, and then that is what's going to exert the effect. So they had to give it a name, calmodulin, calcium modulated protein. So that's what the calmodulin is, the protein that calcium interacts with. When they looked for what's going on, Arabidopsis, again, that's kind of the guinea pig plant of when they do research with plants, 2% of the Arabidopsis genes were turned on. So that's over 500 genes when you touch it. 500 genes getting turned on just by simply touching the plant. And so that pointed to the idea that plants are very responsive to touch, and that it must be something that they need to pay close attention to in order to be able to respond to it appropriately and live their life to full potential. So this serendipitous discovery really led to a lot of other studies that really showed the important role of calcium. Also, the idea that plants put these, well, we'll get into a little bit how they sense the touch, but they're all over the plant body. And it's a very important thing for the plant to pay attention to. So I put this up here not to ask you to memorize it. I'm just showing you that it's complicated at the cell and the physiological level. But basically, this is a diagram trying to show that calcium's involved in getting the signal that the plant is sensing outside, so to speak, and getting that brought into the cells and getting things to shift at the genetic level, shift gene expression. And so they're just trying to show the calcium are the yellow balls in this. And you can see that they're coming in through this protein, the calmodulin. You can see the protein that the calcium is interacting with. So if you just look at this figure in general, you can see there's a lot of yellow balls. Essentially, a lot of calcium is flowing into the cells, getting gene expression altered. You got things going on in the vacuole. There's ATP being used. All right, so it's complicated, and we're not going to get into that. But I'm trying to put a visual in here to show you that calcium is a very important signal molecule, a second messenger for the plant when it's trying to assess what's going on around itself in the environment and then behave appropriately to that stimulus. So we're going to talk a little bit more about touch. We're basically going to look a little bit deeper at some of the plant senses, like we have touch and smell and hearing and taste. Did I say that one? What else do we have? Did I get them all? You know what I mean? Senses. We're going to get into that with the plants too. And so, for example, smell was one of the earlier ones that they were looking at because they had nailed down the idea that plants can release volatile chemicals that can interact with certain receptors in the plant and analogous to the idea of smell. And so if you think about smell in humans, this is how they often define it. The ability to perceive odor or scent through stimuli affecting the olfactory nerves. Olfactory nerves connect the smell receptors in the nose to the brain, and it's a very short connection. If you take that definition and think about plants and rework it a little to say the ability to perceive odor or scent through stimuli and call that the way that plants smell. All right, so that second definition is what we're considering with the plant. Perceive odor or scent through stimuli. And so in this series of studies that they did, this is an example of the dodder plant, Dodder, dodder, not daughter as son and daughter, but anyways, so here is the dodder plant, this little worm-like thing. Dodder plants are parasitic. They have, I think, 24 hours to latch on to a host or they die. And in this case, it's growing near a tomato plant. They've done all kinds of interesting studies where they're looking at what if you give a dodder plant two options. You give it a tomato plant on one side of it, and on the other side, I think they did like ryegrass or something. And the dodder plant with videography sped up, that you could show that the dodder plant was kind of going back and forth between the two, and then very quickly honed in on the tomato and grew toward the tomato and then wrapped around it and started parasitizing it. And so when they were doing these experiments, it's like, I wonder if the tomato versus the rye, are they giving off different volatiles that the dodder is able to perceive? And the dodder knows which one is the better plant to attack because it's more nutritious or it's just better able to attack that particular plant. And so these experiments were done and over and over and over again. And every single time the dodder plant would always go to the tomato, but it would first do that searching back and forth like it was assessing and then go over to the tomato. They even tried to fake out the dodder where they would take the volatiles coming off the tomato plant and put it onto, expose it. I don't know if they put it over a plastic plant, but the dodder was picking up on the volatiles and that's what it was tracking. And so they were able to show that the tomato plant was giving off certain volatiles that the dodder was able to perceive. And then from the parasitic plant's perspective, it's like, I'm going to grow toward that so I can latch onto it and start parasitizing. And so one of the earliest studies that was done was back in 1983, and they did it with tree species. I think it was poplars, or no, willow. These were willow trees. The next one was poplars, I think, and caterpillars. And so they had the caterpillars chewing on the willow leaves, and they could demonstrate that the trees they were using weren't touching each other in any way, whether through roots or branches or leaves. They were no physical contact. And they could show that the neighboring trees were getting their defenses ramped up, even though they weren't getting attacked yet. And so the idea is that the tree that had the caterpillars, the tree that was getting chomped on, was sending off a volatile. If they weren't physically touching it anyway, they drew the conclusion that it's probably a volatile being given off by the attacked tree, and the nearby tree picked up on it and started to ramp up its defenses. Well, when this was published, it was a big thing. I mean, scientists were like, no, no, no. I mean, they were very vigorously against this idea. And so there had to be more studies done. And they did work with poplar and maple. And this time, instead of doing it out in a field experiment, they took it into a growth chamber and kind of a much more controlled environment and ran these studies. And in this case, they didn't put herbivores, they just tore. They physically damaged the plants by tearing some of the leaves. And then, of course, the controls had no damage. And what they could show is that the trees that were getting torn were getting their defenses ramped up because you're damaging the leaves. But the ones that were in the separate walled off containers that couldn't, they weren't sharing the same atmosphere, they did not have any change. So a more careful study where they could physically isolate the air around the plants too, instead of having the plants out in the field where you can't really do that so well. And so when they went into a lab experiment, they were better able to show that, yeah, it really is a volatile that's being given off. And the plants that are getting damaged are sending it off. Neighboring plants are picking up on it. Okay, so this set up a whole nother question. Like, okay, if they're paying attention to VOCs and other chemical signals, are they doing it on purpose? Is it just an accident? What's going on here? So we'll talk about that next time, but we need to think a little bit about that as we work through some of this content.